Okay, so this is the second of the 4 biotechnology PowerPoints. This is going to get a little bit more in depth in terms of sorting out and then copying DNA. So we recall restriction enzymes that we can use to cut DNA with. If we can cut up the DNA from different people or different organisms, then we can compare it. Why? Uses in forensics, medical diagnostics, paternity tests, evolutionary relationships, and even more. So when we comparing cut up DNA, how could we actually compare the DNA fragments, and how do we separate those DNA fragments? Taking a look at comparing the DNA fragments, we would separate them first by size. And how do we separate them by size? Through a gel or a gelatin. Typically what's used is agarose gel. It's actually made from algae, hence the name. And then you do a process called gel electrophoresis, and we will be doing this in class. What? DNA jello? I would not add marshmallows to this. I don't think it's very sweet. So let's take a look at gel electrophoresis and how it works. This is just a way to separate DNA in a gelatin-like material using an electrical field. So we'll be using some electrical fields as well. DNA, recall, is negatively charged. Take a look at your phosphate groups, right? The negative charge. When it's in an electrical field, it would of course move toward the positive end. That should make sense. So here is a side view of your gel box, if you will, and this would be the positive end, and then DNA would run through the gel toward the positive end. Kind of like swimming through jello. See, in your gel, we'll insert some little holes, and then you put your DNA sample down in this well, so it will actually travel through the gel itself. The larger fragments get stuck behind, and the smaller ones are able to travel farther through the pores of the gel. So they're swimming through jello. So again, the DNA moves through an electrical field, and how does that help us to compare DNA fragments? Again, the size of the DNA fragment will affect how far it travels. The smarter, that's a new word, small pieces travel farther, and the large pieces travel slower and lag behind, just like we saw before. Here's some real images of gel electrophoresis, where DNA has been mixed with a restriction enzyme to cut it up into pieces. Then the samples were placed into wells, and then they're subjected to a power source, run through the gels, and then in the end, what you see is this banding pattern. And again, the large pieces will stay nearer the wells, and the smaller pieces or fragments will have migrated the farthest. Then you can illuminate it with a special light to see those fragments even better. So when you run a gel, the fragments of the DNA separate based on size. This is again just a diagram of what we've been talking about. You've got your mixture of DNA molecules of different sizes. This is the cathode is your negative end, the anode is a positive end, and then you put them into your wells. The gel is placed into either a plastic box or a glass plate, and also a solution is poured over the top, a conducting fluid. Then the DNA was cut, remember, with restriction enzymes, and it's placed in here, and then it will migrate various lengths, like so. Then you stain the DNA with lithium bromide, and it binds to the DNA so that you can see it better under fluorescent light, like you see here. Pretty nifty. So let's take a look at some of the uses. We can use it to establish evolutionary relationships, where we can compare DNA samples from different organisms to measure out evolutionary relationships. How related are two different organisms, or more? So if we run your gel, from that, you can create what's called a phylogenetic tree or a cladogram, depending on how you're using it. And if these are our organisms, which correspond with the numbers here, then you can get a sense of the relatedness based on how many bands they have in common. So you can see a turtle and a snake are closely related, and if you look at their banding pattern, they're quite similar. Next in line would be the rat and the squirrel. Their bands are quite similar, which is why they're on the same branch. And then 5 is really not a lot like anybody, so it's kind of your outlier or your oldest one. For uses in medical diagnostics, we can compare the normal allele to a disease allele. Here you have your chromosome with a normal allele and then a chromosome with a disease-causing allele. And this, for example, could be a test for Huntington's disease. And then you run it through your gel. And you can see the variation there. In forensics, we can compare the DNA sample from a crime scene with the suspects and the victim. So from here, you've got the crime scene sample that was taken, the samples from the victim themselves, and then you can compare the DNA from 3 different suspects. And so you want to see where your similarities are. Nothing here for suspect 1, then you have a commonality for suspects 2 and 3, then you see suspect 2, 2, 2, and 2. And so from that, you would probably deduce that suspect number 2 is indeed the actual committer of the crime. DNA fingerprints. Here you can compare blood samples on a defendant's clothing to determine if it belongs to the victim. This is called DNA fingerprinting. Here you'll compare the DNA banding pattern between different individuals, and they're pretty much some unique patterns. And you can take a look here. You'll see the defendant's blood, samples taken from their jeans, from their clothing, and then the victim's blood. So these were the blood taken from the defendant's clothes, so this would be your suspect, and then here's a sample of the victim's blood. So you can see that there are definitely some correspondences in the blood, the DNA from the blood from the victim and from the samples taken from the suspect's clothing. So they match. What about the differences at the DNA level? I thought we all had very similar DNA. How can we use this to actually distinguish between people? Is the DNA different? And if so, why? Let's get back to sections of junk DNA. You might remember these as introns or exons. Think about it. These don't code for proteins, and in fact, they're often made up of repeated patterns, such as CAT, CGG, and others. Now, each person actually may have a different number of repeats, so here is a source of variation that we can use. Many sites on our 23 chromosomes have different repeat patterns, and here's an example there. You see some variation. So let's look at the DNA patterns that we can use for DNA fingerprints. Here's allele 1 in the section of repeats, and here's where you could use some cut sites to cut out that section, and then you cut it, and here you are. Remember your sticky ends. So you run your sample, and there you have the different pieces. Kind of creates a fingerprint. So this would be where this band is. Now let's look at differences between people. So here's that same band from allele number 1. Let's look at allele number 2. More repeats. Notice that the fingerprint is, let me go back, is different between these two individuals. So that's a fingerprint that we can distinguish between. RFLPs. These are called restriction fragment length polymorphisms. Say that 5 times fast. And really what these are differences in the DNA between individuals, and they can be used. So a change in the DNA sequence will affect where and how the enzyme, the restriction enzyme, will cut. So it changes the restriction site. And it will create different fragment sizes and different band patterns. So again, because we have the different DNA fingerprints, when you use your restriction enzymes, you're going to end up with different lengths of fragments. So this fancy word or term, restriction fragment length polymorphism, simply says that the fragments vary in their length, okay? And so when we use our restriction enzymes, you're going to get different lengths of fragments, and so that's how you can tell two people apart. And here is a developer of, or discoverer of, RFLPs and how they can be used. Polymorphisms in populations. We see differences between individuals at the DNA level, and this again is due to the accumulations of junk DNA and how they vary. So here are your restriction enzyme cutting sites, which would result in 2 bands in this case. If you have a single base pair change, then you might get 1 band. If we have accidentally removed this site, now these are fused together. And so notice how the banding pattern will vary in your gel. If you get a sequence duplication, so here, this sequence has been duplicated, you get 2 different bands than we had in the first situation. So these are ways that you get the polymorphisms and then how they can be used. So applying this to forensics, the first case that was successfully used in order to basically show that somebody was not guilty was in 1987. It was a rape case convicting Tommie Lee Andrews and proved that he was indeed not the true raper or rapist. So here's a standard, here's a semen sample from the rapist that was obtained, the suspect's blood, and then a standard again just for comparison. Sorry, the blood sample. And then we see some comparison there. Semen sample from the rapist, different sample. And notice the difference. So in using this information, people can be proven guilty or innocent, and has been used, and we are continually finding out more and more people were put behind bars wrongfully, and in some cases the criminals are still out. Insert quack here. And I have a frozen computer. Hold on, kids. All right, we'll just go back to that slide. That was interesting. I'm not going to start over. Okay, we're on slide 18. Here we go. Sorry about that. So more on electrophoresis use in forensics. So evidence can be used for a murder trial. This is one that you might have heard of before. So we have a blood sample, 3 of them, from a crime scene. Blood sample 1, 2, and 3. Notice the fingerprints. Here's the blood sample from victim 1, from 2, and a blood sample from the suspect. All right. And before I put up the names, take a look at what you got here. Here is a blood sample from the suspect, blood sample from the crime scene, and you can probably see that this one looks like it's going to light up pretty well. Excuse me. With this one here. So let's look at some names. Oh. Maybe you, heard of this case before. So here's blood sample from O.J. Simpson matching up pretty well with blood samples taken from the scene. But as you might remember, he was acquitted. When I was taking some classes at Sac State, one of the genetics professors there actually worked on this case and presented some of their findings on the trial, but obviously that was all ignored. I'm having computer problems. It's frozen. Let's see if it'll get me to the next slide. There we go. Another use of this is paternity testing and who is the father. So here's mom DNA, child DNA, and if one is not certain, here are 2 fingerprints from 2 males. This would be father 1, father 2, and in looking at these you can probably make some judgments about which one it is. There are similarities in both, but the one with the most similarities would be father 2. Now what about making lots of copies of DNA? Remember about our bacteria and how they can copy that DNA for us, lots and lots and lots. But wouldn't it be so much easier if we didn't have to use bacteria every time? Yes, it would. And how can we do that? How can we copy DNA without plasmids? Well, there's something called PCR, or polymerase chain reaction. Remember what a polymerase is? It makes polymers. Chain reaction, you probably know what that is. So this is used for making many, many copies of a specific segment of DNA. You only need one cell to start the whole thing, that cell containing DNA. That means no more bacteria, no more plasmids, no more E. coli smelly looks. They actually really do smell bad. So let's take a look at the process. Really, all you're doing is copying DNA in a test tube. What do you need? Not much. You need a template strand. You need, of course, DNA polymerase, because that's what's going to be making your DNA. And as you might guess, if you're going to make more DNA, you need the monomers. So you need nucleotides, ATP, GTP, CTP, TTP, and of course you're looking at adenine triphosphate, guanine, cytosine, thymine. And a primer, right? We need a primer. Here's a machine that can do this for us. It's called a thermocycler. We do not have one, unfortunately. They're quite expensive. But you will be dealing with this equipment here when we do our transformation labs. So PCR primers. The primers are so important. You need to know a little bit of the sequence in order to make a proper primer, because then the primers can then bracket the target sequence on either end of them. Start with a long piece of DNA and then copy just a specified shorter segment of it. Then you've got primers that define the section of DNA to be cloned, so it's kind of like a starting point. So 20 to 30 cycles of this, and each cycle has 3 steps. It only takes about 30 seconds per step. So PCR is a really versatile technique. You can take a look at this figure while I give you a little bit more information. An important use of PCR now is to actually pull out a piece of DNA sequence, like a gene, for example, from a larger collection of DNA, like the whole cellular genome. You don't want to copy the whole thing. You don't have to go through the process of restriction digest anymore to cut out the gene of the cellular DNA. You can just define the gene with these flanking primers that kind of tag the beginning and end, and get a lot of copies in 40 minutes through PCR and don't have to wait for bacteria to reproduce. But for note, you can also add a restriction site to the copies of the gene if one doesn't already exist, and you can do this by adding them at the end of the original primer, so that's even more efficient. Let's take a look at the process. So what do you need to do? You need to, in a tube, mix DNA, DNA polymerase, the primer, and nucleotides. You need to denature the DNA. You have to heat it so that you can separate the strands, right? 90 degrees Celsius, that is. Then you need to anneal the DNA. And this is, you're cooling it and hybridizing it with the primer. So here you're trying to get it to mix with what you've added into the test tube. So you're building DNA or extending it. So again, the DNA polymerase gets heated to 90 degrees Celsius, the DNA does, so that you can separate it out. But what does the 90 degrees Celsius do to our DNA polymerase? Our bodies don't operate at 90 degrees Celsius. We would not be alive. So we have to deal with that now, don't we? Because we've just killed the enzyme, not that it's alive, that we need. So how do we fix this little problem? We need to heat the DNA to denature it, but the 90 degrees Celsius destroys the DNA polymerase. So we have to add new enzyme every cycle. Well, that's not very efficient at all. It's not really helping us. Not very practical. If you're doing 30 seconds per step, you're not going to sit there and be adding enzyme every half of a minute. So pretty much impractical. So we need an enzyme that can withstand 90 degrees Celsius. Then comes our friend Taq polymerase. This is a hot springs bacteria called Thermus aquaticus. What a nice name. They're very highly thermostable. They can withstand temperatures up to 95 degrees Celsius for more than 40 minutes. And in fact, Taq is patented by Roche. It's a company, and it's super expensive, but apparently there is a black market where people or scientists are selling their own black market Taq clones so that other scientists can grow their own in the lab. Who knew? It's like pirate genes. Kary Mullis is the developer of the PCR technique, and basically it's a copying machine for DNA. He invented this process in 1985, and it really solved this core problem of genetics. How do we make copies of DNA, the strand that we're interested in, but in a timely fashion? The existing methods were slow and expensive and precise, but PCR just turned the job over to the very biomolecules that nature uses for copying DNA. 2 primers that flag the beginning and the end of the DNA stretch to be copied, DNA polymerase that walks along that segment of DNA, reading its code and assembling a copy, and a pile of DNA building blocks that the polymerase needs to make the copy. He later wrote this quote in Scientific American. Beginning with a single molecule of the genetic material DNA, the PCR can generate 100 billion similar molecules in an afternoon. The reaction is easy to execute. It requires no more than a test tube, a few simple reagents, and a source of heat. The DNA sample that one wishes to copy can be pure, or it can be a minute part of an extremely complex mixture of biological materials. The DNA may come from a hospital tissue specimen, from a single human hair, from a drop of dried blood at a crime scene, from the tissues of a mummified brain, or from a 40,000-year-old woolly mammoth frozen in a glacier. Take it from there. So here, if you need to go back and look over anything else, feel free. And if you don't want to listen to me anymore, you can turn off the sound.